So good morning and welcome back to Excelsior Academy. So today we are going to study the second part of amino acids. So all these topics have been divided in different parts with increasing complexity of the topic. So let's begin with our journey. So in the previous class, we talked about different types of amino acid, correct? We talked about different types based on their chemical nature, based on whether it is essential in the body, semi-essential in the body or non-essential, right? And then we talked about based on the R group. So I hope you all have studied its nomenclature of 3 letter and 1 letter. Now, if I say that this is PYQ, it's a tripeptide. So now who can tell me what all amino acids are present over here? Harshita, can you tell me what all amino acids are present? PYQ, it's a tripeptide. So what are the amino acids? Anyone else?
Proline, tyrosine, and glutamine.
Proline. Very good, very good.
Tyrosine.
It is proline, tyrosine, and glutamine. Okay, good. Okay, so now we are going to study about the Zutter ion and isoelectric point. This is very important. So if you are in the research, every time you have to deal with isoelectric point. It is also represented as Pi. Okay. Now, what do you mean by Zutter ion? So whenever an amino acid is dissolved in water, it exists in a solution as a dipolar ion or Zutter ion. It's a Greek word. Okay. So dipolar ion means, see, let us say that this is your amino acid. Can you see two ions? So we have got negative charge and positive charge. Okay. So that is dipolar. So both positive as well as negative is there. So the net charge becomes your 0. Is that clear? Even though the amino acid has got carboxylic acid negative and amino group as positive, it is a dipolar ion having both positive as well as negative charge. So the net charge becomes 0. Okay. Now see, Zitter ion can act as either acid. Acid means proton donor. It means H plus will be released, or as a base, that is proton acceptor, H plus will be taken. So we have got the Zitter ion. Now see the structure. COO minus NH3 plus. So if it is acting as acid, it is going to give the H plus. So from where it is going to give? From here. Okay. Never from here, because over here it is covalent bond, and breaking covalent bond is not that easy. So it is going to give from its amino group. Now if it is acting as a base, that is proton acceptor, then what? Where it is going to get added? Over here. Okay, and nowhere else, because all the valence electrons has been fulfilled. So here this becomes your COOH. Stop me in between if it is not clear. Okay. Yeah. Now, substances having this dual nature are called amphoteric and are also called ampholites. Why? Because they can act both as acid as well as base. Okay. Now, a simple monoamine monocarboxylic acid such as alanine is a diprotic acid when fully protonated. So fully protonated means maximum H plus is present. So it has got the COOH group as well as the NH3 group. So they can yield the protons. So let us see this figure over here. So you have got a monocarboxylic amino acid. It means what? One carboxylic amino group is present. Okay. Now, see over here. So if you have got over this figure is there. If this figure is there, you can see that from where the first H plus will be released, always remember it is from your COOH. So your first hydrogen ion or proton will be released from your carboxylic acid. Now it becomes your total net charge as 0, because minus and plus. Here it was only plus. So you had net charge as +1. Then the second proton will be released from your NH3 group. Then what happens? this becomes negative charge. Okay, and this is very important to understand. If this is clear, then only we can move on to the next. So guys, is it clear to everyone? Yes, is it clear? Because then only in further slides you will understand. Okay. Yes. So before we proceed, just remember what is pH. It is nothing but your proton concentration, okay, which is written as H plus. Now, what is pKa? It is equilibrium constant for ionization. It will be more clear. So it is nothing but the measure of the tendency of a group to give up a proton. So it is a measure of a group. So what are the groups? What are the groups?
CO.
Very good. COH, as well as we have got the amino group. Correct. So these are the two groups, and pKa talks about its tendency to give up a proton. Okay. Now with that tendency decreasing tenfold as a pKa increases by 1 unit. So what does it mean? If pKa increases, the tendency to give H plus increase or decrease? Tell me. If pKa increases, what about the tendency to give protons? Whether it will increase or decrease? It will decrease. Okay, that is to give the H plus. So they are inversely proportional. Getting? It's clear? More the pKa value, less tendency to give H plus. Am I clear?
Yes, ma'am.
Okay. So just understand, because it's all becoming complicated from here onwards. So let's talk about the titration. So this titration we have done all in our school days, right? So it is nothing but it involves the gradual addition and removal of protons. So either you will be adding an acid or an alkali. And then we check the change in the color, right? In that time we check the pH and all, and that's how we determine. So in the case of amino acids also, we are going to study the same. Okay. So here the example has been taken. Can you tell me which amino acid it is? It is glycine, right? So instead of CHH, it is written as CH2. Then you have got COH and NH3 plus. So it is glycine. You have got R as H. Okay. Now, it shows the titration curve of the diprotic form of glycine. Diprotic, why it is diprotic? Because you have got H and H over here. So, two protons are present. Now, the plot has two distinct stages corresponding to deprotonation of two different groups of glycine. So, now we are going to start the amino acid titration curves, in which we are going to talk about how exactly these protons are going to be released. So since we have got two protons over here, correct? So we'll see two changes. Okay. Now see the figure over here. So once you start the titration, you keep on increasing the pH and you start from lower pH to till 12. Okay. Even though on the scale of pH we have got 1 to 14, we do the experiment still pH only. Okay. Now glycine is a, now what is happening? So when you start your amino acid titration, from where the first proton will be released? I have mentioned already.
COOH.
Yes, from here, right? So first one. So you see that, and every time when you start adding, this substrate and this substrate will come in equilibrium. Okay, and that's how your titration works. So you keep on adding slowly until there is a change, right? So in this case also, before that change is happening, all those substrates, that is your, this is the form of diprotic. So what is the net charge over here? Here the net charge is +1. Okay, see, it is given, and here there is no charge. Now what is happening? It is acting as a acid and releasing H plus. It is in equilibrium. Now the net charge is 0. Okay. Second, now second proton will be released from here. Sorry, here. Here the net charge becomes -1 as mentioned before. Okay. Now we see that there are two values. It's called pK1 and pK2. Okay. So this is nothing but your equilibrium constant, that is the tendency to give your protons. Now, the thing is, when you do the experiment, they found that these are the values they obtained. Okay. So what are the values? 2.34 and 9.60. So what does it say about this? That losing proton from carboxylic group has got the pK1 value of 2.34. Okay. Losing proton from amino group has got this value. Clear? This much is clear? Okay. Now, at very low pH, the predominant ionic species to glycine is fully protonated form. Okay. So this is your actually the protonated form. Now, deprotonation means removal of H+. At the midpoint in the first stage of the titration in which COH group of glycine loses its first proton. So this is already just mentioned. Equimolar concentration of the proton, donor and acceptor species are present. Now, this is what we were talking about. So they come in equimolar concentration. Okay. At the midpoint of any titration, there is a point of inflection is reached where the pH is equal to the pKa of the protonated group being titrated. So this one. So pH becomes equal to pKa. For glycine, the pH at the midpoint of 2.34, thus it's from the group has this. Okay. So this is your first. So pK1 is nothing but your first pKa value. So which talks about the tendency of losing a proton from a carboxylic group. Clear? This is your first curve, first part of your titration. Now, as the titration proceed, another important point is reached. It's called 5.97 over here. So this was first, second, and third. Here, there is another point of inflection at which removal of the first proton is essentially complete and the removal of the second has just begun. So which part they're talking about? Over here. So already removal of proton from here, it has been completed. Now, the removal of second H plus from here, it is going to start. At this, pH glycine is present largely as the dipolar ion or the Zwitter form. Okay. Is it clear? I'm going very slowly because it's complicated. Now, the second stage of the titration corresponds to the removal of proton from the NH3 plus group of glycine. At the pH at the midpoint of this stage is 9.6. Okay. It is labeled as pKa for your NH3 group. This was for your COH group. Now, titration is essentially complete at the pH of 12, at which predominant form is going to be your NH2CH2COO minus with the negative charge, net negative charge. Now, if you see the graph over here, what can you tell about these values? What relation do you find among these values? Can anyone tell me? Anyone? So the relationship between pK1, pK2 and pi, what is the relationship? Look at the
Pi is equal to pK1 plus Pi is equal to pK1 plus pK2 divided by 2.
Very good. So this is how you calculate your pi. And what is pi? Your isoelectric point at which the negative charge is 0. Okay. So it's clear. So when you see the relationship between pK1, pK2 and pi, you can actually calculate pi. So pi is nothing but the average of the other two pKa values. Very good. So now, okay, so this is the comparison which is given about the glycine and about the acetic acid having one single carboxylic group and methylamine having NH3+. So these are the standards. So every time when you do any experiment, you always have standard or reference with which you compare your test sample, right? So here in this case, when the titration was carried out, you see that the removal of H+, okay, the pKa value was about 4.8. But in case of glycine, we got 2.34 from again the CH group. Methylamine gave the value of 10.6. But in case of glycine, we got 9.6. So in both the cases, we saw that there was decrease in the pKa value. Now, what is the reason why this is happening? Okay, so this is very important to remember. So this is happening because the repulsion between the amino group and the departing proton, it will lower the pKa for the carboxylic group. And oppositely charged groups lower the pKa by stabilizing the Zwitter ion. So this group is interfering with the removal of proton from the COOH, because of which the pKa value decreases. Okay. Similarly, the electronegative oxygen atoms of the carboxylic group pull the electrons away from the amino group, and because of this steric hindrance, the value from 10.6 it becomes 9.6. Clear? So this is why the value decreases. Is it clear? See, in chemistry, when you study, the neighboring groups are always interfering, and because of the repulsion, the value decreases. Can I proceed? Is it clear? Ranjan? समझ में आया?
Yes, ma'am.
Okay.
Yes, ma'am.
Good. So now titration curve. One second. Yes. Now, the titration curves predict the electric charge of amino acids. So this just now we talked about, okay? So this is nothing but we are talking about the PI value, how to calculate the PI value over here. So see, titration curve of an amino acid gives the relationship between its net electric charge and the pH of the solution. So now we know what is PI. It is having the zero negative charge, right? And then the PI value can be calculated by taking the average of both the pKa's values. So at pH 5.97, the point of inflection between the two stages in its titration curve, glycine is present as its dipolar form. That is your Zwitter ion having no negative charge. Sorry, having no net charge. Now, the pH at which the net electric charge is zero is called isoelectric point. Okay. Now, for glycine, it has been calculated like this, taking the average of both the pKa's value. Now, this is all about amino acids, and amino acid doesn't precipitate. They are soluble form. Okay, they are present in the soluble form. Remember the first slide of the amino acid? So if you add trichloroacetic acid, which is a precipitating agent, all your small molecules like amino acids, fatty acids, sugars, they will not get precipitated. Sorry. Now, if suppose you had a protein, let us say that we have hemoglobin. We know that it has got these 4 subunits. Okay, it's a huge protein. Now, if I add TCA to hemoglobin, what will happen? Whether it will precipitate or not? What will be your answer? Yes, anyone? If I add TCA to hemoglobin, it will precipitate or not? Bhavika, what is your answer?
Yes, ma'am.
It will precipitate. Okay. So what happens? Precipitate means we are adding acid, and it precipitates out because there is a point at which the pH is becoming equal to Pi of the hemoglobin. What does it mean? It means, see, even hemoglobin is a protein, correct? It will have some charge, either positive or negative overall. So when you start adding acid, there is a point at which the pH is going to become equal to its Pi. So Pi of the protein. Pi of the protein means net charge is going to be 0, and that time the protein precipitates out. Okay. Remember this topic because when we are going to talk about protein purification, this will come. Now, glycine has a net negative charge at any pH above its Pi is sorry, okay. So above any the net negative charge at any pH above its Pi, correct? Please mute yourself. Sound is coming. Okay. So glycine has net negative charge at any pH above its Pi. Do you agree? So what will
Yes,
happen? So the negative charge is there. So when you place in the near the electrode, so it will move towards the positive electrode, the anode. Okay. Now suppose the pH below its Pi, it will have the positive charge. So obviously it will move towards the negative electrode or the cathode. The farther the pH of the glycine solution is from its isoelectric point, greater the net electric charge of the population of glycine molecules. Okay. So now this is also very important point to remember about SGS page. Do you know what is SGS page? It is your electrophoresis for to see whether the proteins are purified or not. Okay, so we'll study this also in detail, and here the glycine plays very important role. So just remember this part when we talk about this instrument. The role of glycine will come, and you have to remember the Pi of glycine, so above which, below which, and where it is going to proceed, either to the anode side or to the cathode side. Okay. Yes. Now, amino acids differ in their acid-base properties. What does it mean? Since all amino acids are different because of the presence of the R groups, so their acid-base properties are also going to differ. Okay, so mostly pKa of the COH group falls in the range of 1.8 to 2.4, right? And pKa of NH3 groups falls in the range of 8.8 to 11. Now, what about the R groups? So, if amino acid is having the side chain and which can also be ionized, then it is going to vary. Now you tell me, if I have alanine, how many pKa values will be there? Or if I have arginine, how many will be here? Tell me. Yes, anyone? Sandhya? See, for this, that's why I told you should know the structure. So, for your R groups to be ionizable, you need to have some charges to the R groups. Now, what is the structure of alanine? If this is the, I'll make the basic structure. What R group should come over here?
CH3, ma'am.
Very good, CH3. Now tell me whether you can lose the H from CH3. Can you lose it? Is it possible? No. Otherwise, the valency of carbon 4 will get disturbed, okay? And we don't see like CH2 plus. We don't see like this. So alanine is not going to have the extra, it is going to have again 2 pK values for NH3 plus as well as for COO minus. Okay. Now arginine has got the charge, it is a positively charged amino acid, correct? So it has got the double positive charge. So there you can see 3 pKa values. Similarly for histidine. So all positively charged or negatively charged amino acids will have at least 3 pKa values. The titration curve becomes complex with 3 possible ionization, thus they have 3 pKa values. Okay. Now, let's see for the example. So we have got one example of glutamate. Glutamate is what? Your negatively charged amino acids. Correct? Now see the structure. COOH, this much is your basic structural, and then you have got CH2, CH2, COOH. So now we have got one group from here for proton donor, second group from here, and then third group from here. Okay. Now, as I mentioned before also, which group is going to lose the first proton? It is your COOH. So from here, the first removal of H plus will happen. Okay. So that is the value over here you get as 2.19. Now, second group again. So always it's from your carboxyl group. Then the last one will be your amino group. Okay. Now, second group will be removed from here. Now, removal of this will result in the pK value of your side group. So it is represented as pKr. Next, it will be removed from your NH3+. So hence, finally, we have got pK1 for your first carboxyl, pKr for your second from the side chain, and pK2 from your NH3+. Okay. So we have got 3 pKa values. Now we need to calculate pi. So how are we going to calculate the pi for this glutamate? Yes, anyone? So if you had 2 pK values, you know pi is average of those two. But now you have got the 3. So what will be the answer? So pi is given as 3.22. So what exactly had happened between pK1, pKr and pK2?
Pi for glutamic will be equal to the average of pK1 and pKr, ma'am.
Okay, very good. Now why you have taken these two only? Why not the why not this pK2 and pKr? The answer is correct. Oh, time is running out. So answer is correct. So 3.22, that is your pi is equal to half of your 2.19 plus 4.25. That is the average of these. But my question is why you have taken these two and why not pK2 and pKr? Yes. See, obviously, right? It is because of the same group over here you are talking about, carboxylic group. Correct. So you have to take the average of those two. It's not like you have to take the average of the minimum two. It's not like that. You are going to consider the groups over here. So we know that the first one was carboxyl, second was carboxyl. So the pi is going to be near to that. Similarly, now for histidine also, you have got the values are there, pK1, pKr and pK2. Okay. Now here in this case also, again, so you have the values are, see, here also, if you see the figure, first was removed from here, correct? Second from your group. So where is the group? It is again your amino group of your cyclic chain in the histidine. And then it was removed from the NH3+. So now when you take the pi of histidine, it is going to be the average of pKr plus
PK2.
Very good, pK2. Okay, now it is clear. So in the exam also, they will give you all these graphs and values. So don't get confused like which two values you have to take the average. You need to understand that it has to be the average of the similar groups. Either you take amino or you take carboxyl group. So among all the three, two will be carboxylic or two will be amino group. So if it is given amino, you take the average of 2 amino groups. If it is carboxy, you take the average of 2 carboxy groups. Clear? So this is how you calculate the pi of amino acids. Now to calculate the pi of a protein which is made up of so many amino acids and having so many side chains. So how do you calculate? For this, we have got something called isoelectric focusing. Okay, it's a technique. So we are going to do all this thing. I'm just mentioning over here. So pi of protein is calculated using isoelectric focusing. Okay. Yes, thank you, people. So just let me know if any doubts are there.